Hello, everybody. Welcome to this week's lecture. My name is Andrew Serban, and today we will be discussing enzymes and their role in the body. Before we begin, I want to start with a simple thought experiment and prove my point about why enzymes are important in our daily life. So if we look at sugar, table sugar, which most of you have in the pantry, if you leave that sugar in a bag and you just let it sit there for days and days and days and months, nothing will happen. It will remain sugar, and you will get no energy out of that sugar. Now, if you take sugar and add in some kids, or feed it to kids, for those of you who are parents, you know very well what happens. You get too much energy, right? So something obviously is happening when we feed it to kids, or any adult for that matter, or any biological organism that can metabolize sugar, in which we are able to extract energy from that substance. And so part of the question is, how does this happen? And we'll find, hopefully today, that enzymes are a key role in turning sugar into energy. So energy is a big part of our discussion, and if you guys recall from your general chemistry courses, thermodynamics is basically the study of how energy flows. Cells are governed by the same laws of physics and chemistry, and so as we think about the flow of energy within a cellular environment, thermodynamics laws apply. So if we were to take a macro perspective of energy flow, right, energy flow in the biological world looks something like this. You start with energy from the sun, so photosynthetic organisms can capture that energy. They store that energy as potential energy inside the chemical bonds, right, inside the plants. And subsequently, animals or humans or whatever eats those plants can break down those bonds that were generated by the organisms and extract energy from them. So this whole idea of energy flow is very important in understanding how organisms work. Now, tying back to your Gen Chem days, redox reactions, which hopefully sound familiar, are exactly that, creating neutral states into lower and higher energy states, right? So oxidation, if you recall, is an atom or a molecule that loses an electron. Reduction is the exact opposite. It's the gaining of electron. And so these redox reactions are paired, but you generate something that is either lower or higher energy as a result of this reaction. So to refresh your memory on Gen Chem, the first law of thermodynamics is that energy cannot be created or destroyed. Energy can only change from one form to another. The total amount of energy in the universe remains constant, and during each conversion, some of your energy is lost as heat. So first law, energy cannot be created or destroyed. The second law of thermodynamics is the law of entropy, or disorder, is continuously increasing. So energy transformations proceed spontaneously, and they convert matter from more ordered to less ordered forms. And an example of that would be ice, right? If you put ice in a glass with water, over time that ice will disappear, which is tending from more ordered to less ordered structure. This is relevant because the main goal in understanding enzymes is to understand this idea of free energy, right? So if we look at this formula here, we have G, which is the energy available to do work, H, which is enthalpy. Enthalpy is defined as the energy in a molecule's chemical bonds. T is the absolute temperature, S is entropy. We can start ascertaining what is actually happening with the free energy of the system. So if we look at the changes in free energy, right, we can have a positive change. So if we have a positive delta G, if you recall, is the change in free energy. A positive delta G looks something like this. The product of the reaction has more free energy than the reactant, meaning that H is higher or S is lower. It's non-spontaneous, meaning it requires energy to be added to the system, right? Conceptually, if we think about that, it makes sense. To go from reactant to product, you need some sort of energy boost to get you from the reactants down here to the products up here. So you need to add energy into the system, meaning your delta G is positive. The opposite would be a negative delta G, where the products are actually lower energy than the reactants. And you could imagine you don't need energy being added to the system to drive that reaction. In fact, you get energy being released as a result of that reaction going forward. And so with a negative delta G, that is a spontaneous reaction, or termed exergonic. Now, if we were to plot it a little bit differently and look at the reaction over time, you would see something like the blue line. You start with your reactant, you have a hill to climb, and when you get to the top, you have a downward trend towards product, right? So you need some energy to get up, but then once you're there, you release some energy. And so if you're looking at the, let me get my pen here. If you're looking at the change in energy, it would be this bar right here, which is going from this state to this state. So this release would be your delta G. Now, one thing you may notice is this hump right here. So from where we start and to where we need to be, this is called activation energy, okay, here in blue. So this activation energy tells us the amount of energy that we need to overcome in order to get up to where we need. Now, you may recall from Gen Chem that you can have either an uncatalyzed reaction, which is perceived here in the blue, or you can have a catalyzed reaction, which is here in pink. Now, if you add a catalyst to your system, you reduce your activation energy significantly. So instead of this red bar here, you would have an activation energy that looks like this, approximately. They should overlap, but I drew it a little staggered so you can see for clarity. You would have a much lower activation energy, which means you need less energy to drive your reaction forward. Now, if we were to define activation energy, it would look like this: extra energy required to destabilize existing bonds and initiate the chemical reaction, right? And that is driven by a few things. Chiefly, the larger the activation energy, the slower it goes. That makes sense. But you can increase it by increasing the energy of the reacting molecules. You can heat it up, or you can lower the activation energy. That's how we can get past it. And so you add the catalyst, right? Substances, just defining it here, a catalyst is a substance that influences the chemical bond in a way that lowers its activation energy. But importantly, it does not alter the proportion of reactant turned to product. So it's independent of the reaction, meaning it doesn't get consumed within the reaction. Now, this same concept of catalyst driving a reaction forward and giving this release of energy applies to biology as well. As it turns out, there are some things called biological catalysts, or as you know them, enzymes, that can do very similar things and increase rates of reactions within the cell. So giving a brief history on these biological catalysts, we look to the 1850s, which is quite a long time ago. Louis Pasteur, he discovered in the 1850s that yeast cells were actually responsible for causing the conversion of sugar to alcohol. So he said, okay, he did this experiment converting sugar to alcohol, and he realized that it's the yeast cells that do it. In the 1890s, Edward Buckner actually made a second discovery, in the sense that he realized it wasn't the cell itself that caused this conversion, but rather the contents within that cell facilitated the conversion. These contents were later called enzymes by Frederick W. Kuhn. So as we start thinking about enzymes and we define them, most enzymes are proteins. General rule up until about the late 80s, early 90s, most enzymes were all proteins, but we've recently started discovering that some actually can be RNA. And discussing RNA type enzymes will not be the focus here, but just understand that not all enzymes are proteins, some are RNAs, but the bulk of them are. Understanding enzymes is key in terms of their shape, because their shape stabilizes the temporary association between the substrates. And enzymes do not change or are not consumed within the reaction, right? So it is a true catalyst in that definition. And here's an example of what an enzyme could do within biology. So carbonic anhydrase, if left alone with no enzyme, you would have 200 molecules of carbonic acid generated per hour without the enzyme. If you add in carbonic anhydrase, you have 600,000 molecules formed per second with the enzyme. So the addition of this catalyst drives the reaction forward exponentially. So enzymes are very key in facilitating fast reactions in biology. Now, if we were to give a cartoon representation of a general enzyme, it would look like this. So the blue piece of the diagram would be the enzyme itself, and in yellow would be the substrate that binds in it. Now you notice the shape of the substrate mirrors the cleft, or the active site, if you will, of the enzyme. And so you have the substrate binding to the enzyme inside the active site, generating this enzyme substrate complex. Now, if we were to take a closer look at the active site, it is the pocket for the substrate. It forms that enzyme substrate complex, and it is a precise fit of that substrate inside the active site. Okay, it applies stress to distort particular bonds and lower the activation energy, which gives it this induced fit. Now, if we were to look at a cycle of it binding and unbinding, it would look something like this. So we can look at sucrase, for example. Sucrase breaks down sucrose, which consists of glucose and fructose. Those are two types of sugars that you may have heard of. So we have the enzyme here in this state being completely empty with its active site. It comes into contact with sucrose. Sucrose binds to the enzyme in the active site. The bond is then broken here in the middle, generating glucose and fructose. Those products dissociate, yielding an empty enzyme. And this cycle can start over and continue over and over and over. So this would be an example of a cycle of how an enzyme works. Now, what is interesting about enzymes is that their rate depends on two key factors. Okay, so you can imagine, go back here, the more sucrase we have, the faster this reaction will go, or the more glucose and fructose you will generate, right? You have more and more and more fructose and glucose being generated, right? So concentration is key. The higher the concentration, the more product that you will get within the time frame. But then another key piece to understanding their function is all of the chemical and physical conditions that the enzyme is within. So for example, if we look here at temperature, temperature has an effect on activity, or the rate of reaction. If we look at this human enzyme, for example, activity at 30 degrees Celsius is much slower than activity at, let's say, 41 degrees, right? If we look at a hot springs prokaryote, right, it's sitting in hot springs thermal water, we expect its activity to be optimal at those high temperatures, and sure enough, that's what we see. At 55 degrees, activity is pretty low. At 72 degrees, it's pretty high. Okay. Now, another factor is pH, right? In Gen Chem, we learned about pH and what that means for the concentration of hydronium ions. Well, it turns out that can have an effect on the enzyme function as well. If we look at pepsin, for example, pH of 2.5 is optimal for pepsin, but trypsin, which is another enzyme, has an optimal pH at around 6.5, right? So as we think about activity and function, we need to be cognizant of concentration as well as the other factors in the environment. Now, enzymes do have inhibitors that can bind. So we looked at having a substrate bind, but it turns out an inhibitor can bind as well. That can decrease the activity. So an inhibitor is defined as a substance that binds to an enzyme and decreases its activity. There's two types. We have competitive inhibitor, meaning that it competes for the same binding site or the same active site as the substrate. And if it binds first, then that substrate cannot bind in that site. But we also have non-competitive inhibitors, meaning that the inhibitor will bind to the enzyme at a different site than the active site. And this binding to a different site causes a shape change within the enzyme, and it can no longer bind its substrate. So let's look at an example of that. If we look at this figure A, you'll notice that the substrate here in orange would have fit beautifully inside the active site. But you have this green molecule that binds, that's an inhibitor, that beautifully takes up this particular space and prevents the substrate from binding. So that would be a competitive inhibitor. Now, if you look at the panel on the right, panel B, you will notice that the inhibitor is actually not binding anywhere near the active site, but rather it's binding in a different site that causes a unique shape change here. Okay? This shape change prevents the substrate from docking appropriately and therefore cannot bind. Both inhibit the enzyme, but they do so in different ways. Now, to add a little bit more complexity to enzymes, we have what are called allosteric enzymes. These enzymes are defined as enzymes that exist in this active and inactive form. If we break it out a little bit between we can differentiate a little bit between non-competitive inhibitors and allosteric inhibitors. So non-competitive inhibitors, as we just mentioned, bind to that allosteric site, and it's an on-off kind of dynamic. It binds, it turns the enzyme off, and the substrate can't bind. If you look at an allosteric inhibitor, though, you have an inhibitor that, when binding, changes the activity. So it doesn't necessarily turn it completely off, but it can reduce its activity to some extent. Similarly, with these allosteric enzymes, you have what are called allosteric activators, and these can bind to an allosteric site and increase the enzyme activity, right? So this would be on-off. This would be limiting its activity, okay, and regulating its activity. Now you may ask, why is this important? Why do we care? In isolation, you take sucrose, you put it in a vial with sucrase, it generates your fructose and glucose, and you're good. But in biology, we care about regulating and fine-tuning that process, right? We care about the biochemical pathways. So let's look at, for example, a biochemical pathway within an example cell. Okay, so reactions occur in a sequence, right? The product of one reaction is the substrate of the next, and many steps can take place within specific organelles. So let's look at this figure here. We start with an initial substrate. This will bind to enzyme 1. Okay? Enzyme 1 will then turn over and generate intermediate substrate A, which then goes to enzyme 2, generating intermediate substrate B. This intermediate substrate goes to enzyme 3, generating yet another intermediate substrate C. That substrate goes to enzyme 4, generating the end product. Okay? So we care about this regulation because, one, you may have a few different enzymes that are in sequence that depend on all of the different sequences happening in order. But there's yet another example, and that pertains to feedback inhibition. If you could imagine the scenario going to, for hours, it would stop when you have no more initial substrate. That's what clearly prompts this one. But what if we don't want to deplete this entire initial substrate? What if we want to get to a point where when we hit a certain concentration, we want the entire reaction to stop? Well, that's where feedback inhibition comes into play. So the end product of the pathway increases in concentration as it is synthesized, as we saw. You go from orange ball to purple hexagon. So you generate more and more of this purple hexagon, but the more product that is increases the probability that it binds to an allosteric site of an enzyme in the pathway, causing it to change and not bind the normal substance. So in essence, it will shut down the pathway of the raw material and energy not being wasted. So let's look at an example. Same structure here. We have the initial substrate, goes to enzyme 1. The intermediate substrate goes to enzyme 2. The intermediate substrate B goes to enzyme 3. Okay? When we generate the end product now, that end product goes to enzyme 1, which changes the binding site or the active site and no longer can accommodate the initial substance. And so now you turn off the entire pathway. Okay, this is very, very important in biology because we understand the power that enzymes have and the power that they can bring to a cellular environment, but we also know that if we don't regulate them effectively, we can overexpress a particular end product. So enzymes have this capability, but there's all kinds of regulatory components to enzymes. So in summary, enzymes play a critical role in breaking down molecules inside of our cells. They significantly increase the rate of reaction by lowering the overall activation energy required. They're subject to high regulation to prevent overactivity. We have competitive and non-competitive inhibition, as well as feedback loops. And many diseases arise from underregulation or overregulation of certain enzymes within the body. Cancers would be a great example of this. If you upregulate specific enzymes targeted at replicating DNA continuously, that's going to lead to the growth of certain tumors, perhaps, or different types of upregulated components, which is what cancers are. And so much of the research being done is looking at what enzymes within the cellular environment are being upregulated in certain cancers and how can those be downregulated. So hope you enjoyed this talk on enzymes, and I look forward to discussing other aspects with you further. Thank you.